Thank you. So good morning, everyone, and welcome to the Industrial Biotechnology Innovation Catalyst webinar for January. It's our first of the new year. If you've not joined us before, these webinars run every month as a lunch and learn for anyone interested in industrial biotechnology. Upcoming ones are advertised in the university events page, and I think a member of the team will probably post a link in the chat in a few minutes so you can have a look and see what's coming up. For those of you that don't know me, I'm Sabina Hawthorne-Thwaite. I'm the Programme Manager for the Industrial Biotechnology Innovation Catalyst, or IBIC as we call it, which is a place-based industrial biotech impact acceleration account that's funded by EPSRC. We've got £5 million to spend over 4 years. So through the money, we're creating an industrial biotech ecosystem across Greater Manchester, Liverpool and Cheshire to accelerate our research out of our universities in the Northwest, working with industrial biotech industry sectors to create impact on our Northwest region. So the project is led by Professor Aileen Miller at the University of Manchester, and we partner with universities in the region, Liverpool, Manchester Metropolitan, Salford, and Liverpool John Moore University, which brings me nicely to welcoming Dr. Femi Olorunniji and Alexandria Holland, I think, yeah, Alexandria Holland, hi. And they're going to be talking about repurposing serine integrases as tools for engineering biology this morning. So I will pass you over now to Femi. I'll stop sharing my screen so that we can share Femi's slides, if that's okay. Away you go, Femi. Thank you.
Right. Thank you, Sabina, for giving us a nice introduction, and thank you, everyone, for joining us today. The first part of the webinar will be delivered by Alex, who is a research fellow in our group here in Liverpool. Alex did her PhD in Bath, University of Bath, and first postdoctoral research work was at University of Nottingham. So she's been with us now for about 2 years, helping us in working out how to repurpose serine integrases. So Alex will start by giving a broad introduction. Then after that, I'll talk about the specific work we've been doing here in Liverpool. So over to you, Alex. Thank you.
Hello. I'll just share my screen. Okay. Hello, I'm Alex. As Femi said, I'm a postdoc working in this group at Liverpool John Moores, and I'd like to give you an introduction into large serine integrases and how we can repurpose them as tools for engineering biology. Let me just get a laser pointer up. Okay, so what are large serine integrases? So large serine integrases, they are enzymes produced by bacteriophage, and what they do is they mediate site-specific recombination. They're essential for the integration and excision of phage DNA into bacterial genomes. So I'd like to talk you through the life cycle of the phage. So here we have a phage, an infecting phage that's found a target bacteria, and it's injected its DNA into the host. At which point the large serine integrase genes are expressed and the large serine integrases are produced, and this allows for large serine integrase mediated integration of the phage genome into the host genome. So this all happens in the lysogenic cycle of the phage, and this is where the phage is now integrated into the host genome, and it's now known as a prophage. So the prophage can actually remain dormant, and it's actually inherited by all daughter cells during replication. But in the event of an environmental stress or some trigger, the prophage then induces expression of both the large serine integrase and also another enzyme called an RDF. And I'll talk to you more about the RDF shortly. So once this happens, the phage now enters the lytic cycle. In the lytic cycle, the phage genome is excised, and this is mediated by both the large serine integrase and the RDF. And this allows for the production of new phage, at which point the phage bursts out of the cell and cause the lyse to cell, releasing the progeny, and the cycle starts all over again. So understanding the native role of the large serine integrase gives us insights, and we can also lay the groundwork for their use in synthetic biology and genetic engineering. So these enzymes, they recognize specific attachment sites on the phage genome, known as @P, P for phage, and @B, so B for bacteria. And the recombination event, they create hybrid sites known as @L and @R, left and right. And this signals successful integration. So this is a highly precise and efficient process and ensures the stable maintenance of the phage genome within the host as a prophage. So before I tell you more about attachment sites, let me tell you more about large serine integrases and how they're structured. So large serine integrases, or LSIs, or I would just refer to them as integrases during this talk, just because it's shorter. So I'd like to break down their structure and illustrate how they interact with their target attachment sites. So they are highly modular, and they're comprised of 4 distinct functional domains. So here at the N terminal, we have the catalytic domain. This is defined by its defining feature of having a serine residue at the catalytic site, hence the name large serine integrase. Next is the DNA binding domain 1, also known as the recombinase domain, so DVD1. And now we have a DNA binding domain 2. It's also known as the zinc domain in the literature. And we also have the coiled region. So here I've just got a cartoon of how the domains are arranged. So here we have the coiled domain that sticks out of the DNA binding domain 2. So the process of integration or recombination, this begins with the integrase recognizing and binding the 2 distinct sites on the DNA. So individual large serine integrase monomers, 1, 2, they bind the @P and the @B sites as dimers. They then form a synaptic complex, whereby they become a tetramer, and @P and @B sites come together. This is mediated by the interactions in the coil domains, here in blue. And each integrase catalytic subunit will cleave each one of the 4 strands of DNA, resulting in overhangs around this central dinucleotide region. So now this is called P and P prime and B prime, and what they do is they then switch positions by rotating 180 degrees, and the DNA strands are then religated, resulting in new attachment sites @L and @R. So here I'm showing you one of the only solved crystal structures of an integrase interacting with DNA, which is the L1 integrase, showing how the 2 different DNA binding domains interact with DNA. With the @P site. So it's important to understand their modular structure and to give us insights into how they interact with the DNA and how they achieve their specificity. So now let's talk about @B sites. So phage utilize @P, so which is a fixed attachment site within their genome, to integrate into the host at the @B site, which is shorter. I'll talk about this a bit more. Once inside the host, the phage will then scan the whole bacterial genome for a sequence resembling the @B. And this ensures site-specific recombination, and it's precise. So the attachment sites are typically short sequences, about 40 to 50 base pairs long, with an @P is often longer than @B. And structurally, these sites contain inverted repeats. So what I've done is I've just flipped it over on itself just to highlight the inverted repeats. And they flank the essential dinucleotide. So this is a crossover point, and it serves as the recombination site. So functionally, the length difference, you might be wondering, so it does serve like a functional purpose. It actually influences the conformation of the complex. So as you can see here, when it's bound to the @B site, it's kind of more scrunched up together. And this ensures not only this site-specific recombination, but it also contributes to the unidirectionality. So in this conformation, this will always favor integration over the excision, unless an RDF is present. As I said, I'll talk to you about RDFs more shortly. And when an RDF is present, this induces another conformational change. So how do we know what the attachment sites are? So we can identify potential prophages within bacterial genomes by looking for phage-related genes, such as the genes for large serine integrases. So once we've identified these phage-related genes, we can identify where the prophage is, and then just analyze the flanking sites, the flanking sequences, and from that we can determine the @B locus. So I've mentioned RDFs a couple of times. So as a reminder, they are involved in the excision of the prophage from the bacterial genome. So RDFs are recombinant directionality factors, so RDF, and they're small accessory proteins that interact with the integrase, and they facilitate the reversible, the excision, essentially. So they do this by recombining @L and @R back to @P and @B. So it is essential for the excision step, and as I said, it will alter the conformation of the large serine integrase DNA complex, allowing for the conversion of @L and @R back to @P and @B. So without the RDF, the integrase will be locked in the integration phase. And also something to note is RDFs also inhibit the @B and @P reaction. So when the RDF is present, it can't, the integrase can't take us back to @L and @R, if that makes sense. So if we compare this to something like a tyrosine recombinase, that can catalyze bidirectional reactions. So unlike large serine integrases, RDFs have no sequence homology. So here I'm just showing you 3 examples of known RDFs. These are the AlphaFold models. And they have no sequence homology, no structural homology. They don't look the same, and they also don't function in, they don't have similar functions. And something else to note is that RDFs bind the integrase and they do not bind the DNA, unlike in a tyrosine recombinase, when the RDF binds the DNA and forms a complex. But yeah, excuse me. So something else that I'd like to mention is we've come incredibly far. Phage were first discovered 100 years ago, about, maybe? Yeah. And they are known to be one of the most abundant organisms in the biosphere. So up to 10 to the 31 different phage. And so this is a huge pool for us to work from. And advances in genomic and metagenomic sequencing have enabled the discovery of large serine integrases at an unprecedented scale. So I'll give you an example of this Nature paper recently published a couple of years ago. They mined about 2 terabytes of data, and from that they predicted over 25,000 large serine integrases. So we think that's super cool, because imagine having like 25,000 different restriction enzymes to play with their own cognate sequences. So this is quite exciting, we think. So why use large serine integrase recombination, and why use large serine integrases? So large serine integrase recombination is unique in several ways. Firstly, they are unidirectional. So they catalyze the DNA rearrangements in one direction, unlike, for example, Cree locks, where the unidirectionality is dependent on specific arrangements of the lock sites. They are orthogonal, so that means that they can operate simultaneously in the same system without interference, because they have unique cognate attachment sites, which for us, for example, this can enable parallel assembly of genetic pathways or circuits. They are specific. So they, as I said, they recognize specific attachment sites, and so this ensures precise integration or excision, and this will reduce off-target effects. So if we compare it to something like CRISPR, for example, specificity really does depend on guide RNA and the genomic context, and there's always a risk of unintended cuts. So with large serine integrases, their specificity reduces off-target effects. Also, the reactions are editable. So with the addition and the removal of the RDFs, we can take the reaction, either an integration reaction or an excision reaction. So yeah, they're editable. And they're also efficient. So large serine integrases are incredibly efficient. Most characterized large serine integrases can drive the DNA recombination reaction to almost 100% completion. So I'd like to give you an example of the different types of modifications that large serine integrases can do. So they can enable different types of precise DNA modifications or recombinations. So let's start with the most obvious one, which is an insertion or a deletion. So here I've just shown you like an example of, say, a donor plasmid, or with a gene of interest or a pathway of interest, and we have our target genome with the attachment site. And what this does is it integrates the whole plasmid or our fragment of DNA into our attachment site in our target genome, and the result is a stable insertion. Another example is they can mediate inversion reactions. So depending on how the attachment sites are oriented, so in this case, they're oriented facing one another, when recombination occurs, this will result in an inversion. So this setup can toggle between two states. So say we have a gene of interest or a promoter or something, depending on if we have the integrase or the integrase and the RDF, we can toggle between two different states, for example, in a genetic switch. And another one that I'd like to mention is intermolecular recombination. So in this example, I'm just using a single pair of attachment sites. So we can join two fragments of DNA. And one important thing to note is we can do this sequentially using different integrases and their corresponding attachment sites. So, and this will enable us to, assemble complex pathways. So something important to note is one of the most powerful aspects of large serine integrases is that depending on if they are alone or with their RDF, we can toggle between DNA states and change the configurations in a dynamic way. So I'd like to give you an example of using large serine integrases to assemble a pathway. So here, some work done in Femi's group. They used a technique called SIRA, so serine integrase recombinational assembly. So they used this to assemble the beta-carotenoid pathway. So here, they generated a bunch of fragments using PCR, and these fragments were flanked by attachment sites. So each fragment were encoded either a gene in the pathway or the origin of replication and the, I can't remember the word now, selection marker, sorry. So each cassette was generated and they utilized 6 orthogonal large serine integrases. So this was done in vitro in one pot, and the result was an intact plasmid. The plasmid was then transformed into E. coli, and they showed successful expression of the pathway, and we have our beta-carotene as a result. So something to highlight here is the orthogonality, which ensured that each integrase acted exclusively on its own attachment site, enabling this assembly of this whole pathway. So I'd like to almost finish off with a few challenges that we face in developing large serine integrases. So the number one challenge is identifying RDFs. So the RDFs, as I said, they are essential for controlling the directionality, and they allow for reversible DNA modifications. But I also said that they lack sequence homology, and because of this, they're notoriously difficult to identify. So dozens and dozens of large serine integrases are characterized, but only a handful of them of the RDFs are known and characterized. So without them, the applications are limited to just the forward reaction. So one reaction. without the RDF. So this is one of the challenges that we face. Another challenge is the limited structural insights into the DNA interactions. So the large serine integrase DNA interactions. So we lack we lack high resolution structural data. And as a result, designing or engineering them, large serine integrases that is, remains a challenge. So as I said, this is one of the only structures of the complexes available. And finally, another challenge is specificity. So I say that it is a double-edged sword. So they are highly specific. They only act on the cognate attachment sites. So the specificity is great, but it does limit our flexibility when targeting non-cognate sequences in target genomes. So therefore, expanding the toolkit is super crucial. And tools like AlphaFold, AlphaFold Multimer, they are revolutionizing the discovery and characterization and design of new large serine integrases and RDF identification. And Femi will talk more about that coming up. So I'll leave you here, and Femi will expand more on the work that we're doing in his group. Thank you.
Thank you, Alexandria. So if there's anyone on the call who would like to ask Alexandria any quick questions before we hear from Femi, please do use the raise your hand function, or you can type things into the chat. Speak now, or we will move on.
It might be that we can take questions at the end, maybe?
We could take questions at the end, absolutely, yeah. Okay, Femi, away you go, thank you.
Yeah. Are you able to see my slides? Yes, okay, thank you. Yeah, thank you, Alex, for giving a very excellent introduction. So making it a lot easier for me to go through some of the points some of the things we've been doing in our lab. So I suppose the place to start is to kind of address the key questions we are trying to solve in our group, and one of which is, what is the mechanism? How does integrase catalyze its reactions? Alex mentioned that the reactions are reversible, the integration pathway, as well as the excision pathway. So how do you regulate that? And of course, Alex highlighted the challenge of not having a full structure. So if you don't have a full structure, your ability to design experiments, to test models, or to engineer the system is highly limited. And we as promising as serine integrases are, if we don't have the answer to these questions, invariably we are going to be struggling to take them to the level at which we want to take them to. So the theme of our discussion that says repurposing large serine integrases for engineering biology, that's where we are working toward, and we have some collaboration with local biotechnology companies, even in the Northwest here. So to a large extent, we are moving closer to taking integrases from the bench onto the industrial setting. But for the rest of this talk, I'm going to be telling you some of the points, the things we've been trying to do. So Alex identified a few challenges that we had to deal with. And the first one I want to talk about is, how do you find an RDF? So as Alex mentioned, thousands of integrases, over 25,000, identified in a recent genome screen. Now, you kind of think, if you have such powerful and versatile tools at your disposal, you can do quite a lot with it. And the reason is, it's very easy to identify serine integrase. So they have what we call the characteristic recombinase domain. So if you search in the genome of a phage or a prophage, you can easily tell what whether you have an integrase or not. Now, in contrast to that, the RDFs, they're a different set of beasts, all sorts of random sequences and structure. So that makes it very difficult to find. And of course, if you don't have the RDF, you are limited. Now, if you were working with a tyrosine integrase, it's very easy to find the RDF because they are usually in synteny or in proximity to the integrase. But quite often, as you will see illustrated in this image, the RDF could be anywhere in the phage genome. So our first challenge is that if you don't have the RDF, there is only so much you can do with the integrase. We can go in one direction, but taking it out, the kind of flexible editing we would like to do, is almost impossible. So that's the first challenge we thought we could address. And Alex mentioned earlier that the AlphaFold range of tools, AlphaFold 2, AlphaFold Multima, and AlphaFold 3 have significantly made things a lot easier for us. So what we did was to kind of try to model the structure of the different RDFs that we know. So we used RDF, we used AlphaFold to predict the structures of these RDFs. And then we used AlphaFold Multima to model the non-RDFs interacting with their respective integrases. And two things come across quite immediately. The first one is, as we expect, the RDFs illustrated here in kind of golden yellow have different structures. Though the integrases they interact with are highly conserved proteins, the RDFs have different structure. And when we use AlphaFold Multima to model interaction, then it becomes clear that despite the diversity or the different ways in which they fold, they all interact with the integrase at the same location in their respective integrase. So we call that like a hotspot. And the cartoon I have here shows our integrase, the catalytic domain, the first DNA binding domain, or DBD1, and the second one, and the coil-coil domain. So the pattern coming through in all cases is, despite the structural diversity, each RDF is interacting with its respective integrase at the junction of the second DNA binding domain and the coil-coiled motif. Now, that's a very kind of unusual characteristic. So we thought that's interesting. We had better try to verify if this is genuine. So of course, what you do is that you take your integrase and take the RDF and carry out selective mutation. So we made mutations along the integrase RDF interface with the expectation that if the interface we've identified are functionally relevant, mutations will disrupt your kind of at R by at reaction, the excision reaction. And that's exactly what we did. We find mutants of the integrase and RDF along that interface, which verify the fact that the regions we've identified, the interface, are functionally relevant. Significantly, those mutations did not affect the integration reaction, where you don't require an integrase RDF interaction. So that's the first kind of encouraging thing, that the predictions made by AlphaFold and AlphaFold Multima can actually be validated experimentally. And this kind of data has recently been accepted in nucleic acid research. So having verified the way RDF interacts with integrase, then we go back to the question, can we use this approach to find what protein serves as the RDF among a number of different proteins that are present on the phage genome? So the point we're trying to illustrate here is that if you look at the genome of a phage, there are scores, sometimes 100+ genes, which any of them could be the candidates for the RDF. And the traditional way we will do this is to take out each gene, isolate it, clone it, and do like a 2 hybrid yeast assay to find out if it interacts with the integrase and if it can serve as an RDF by activating the excision reaction. Now, you can easily understand that that's quite an expensive process, it's time-consuming, and quite a lot, you get several false positives. So we went back to observation that if the RDF, irrespective of its structure, is binding to the integrase at a specific location, can we use this approach to streamline the process? So we thought we'll use AlphaFold Multima to screen the genome of a phage and ask which of these proteins will interact with the integrase as a way of identifying the RDF. So we call that virtual pulldown. But for that to work, the first thing we did was to do an analysis on phage genomes of all known integrase RDF pairs, about 13 or thereabouts. We thought we'll take all the proteins in the genome of this phage and ask if this process can pick the right one, which we know. And in all cases, the virtual pull-down picks the confirmed RDF as the top candidates. And what you see here is an example of the kind of data you generate. So the crucial factor here is the IPTM, the blue trace, which is an indication of the interaction between the base, which is the integrase, and each specific protein in the phage genome. So the example here is from phage A118, and in all cases, the topmost mark is the RDF. So having validated that using the different ones we know, we thought, okay, we'll give it a go. But we do need an in vivo reporter assay. So the in vivo reporter assay we use is actually a very cool assay. So essentially what we have is a bacterial construct where you have a red fluorescent protein gene and a green fluorescent protein gene, and you have a promoter that in the ground state is facing the RFP. So if you express these constructs, the default state of the cell is to express GFP. Now, the interesting thing is that the promoter is flanked by an integrase at P site here and an at B site. In inversion orientation, which Alex mentioned earlier, results not in a deletion, but a switch in the orientation of the promoter. So if your recombinase or your integrase is working, it will cut the P and the B site and flip the promoter in the direction of GFP. And then you stop making RFP and start producing GFP. So that's a very easy assay. You set up the reaction, do your transformation, come back the next day, your plates either red or green. Okay, so I just want to mention briefly that this approach too is also very effective in designing a genetic toggle switch, which can be used in regulating cellular events. So having established our virtual pull-down and established an in vivo assay, we thought we'll have a go at an integrase whose RDF is not known. So we started with this one called NM60 integrase. And just as expected, the RDF that the virtual pull-down predicted was able to catalyze the conversion of ATEL substrate to at P and at B, and was also able to inhibit the reaction of at PB to ATEL. So the key point to notice here is that if your RDF is functional, it will convert, it will activate ATEL by ATEL recombination and inhibit at P at B recombination. So the very first example we tested shows that the virtual pull-down works and we can experimentally validate it. Then, of course, the next question is, it works on one, how reliable is the system? So what we did is to try and characterize a few other ones. Now, just to mention that using the traditional genetic approach, over a period of about 20 years in the lab, different groups have only managed to identify 13 RDFs. But within 3 months of establishing this approach, we streamlined the system and over 75 new RDFs were characterized within 3 months. Now that's transformative. Of course, any new system like this, you are going to find false positives. So, and we realize that on the average, 1 in 7 turn out to be false positive. An example here is CB16. For reasons we don't know, it just didn't work in the lab, but the virtual pull-down identified it as an RDF. Now, since these experiments were done, we've had new structures that allow us to do additional structural modeling, revealing that there is a critical interaction required to be an RDF, which our virtual pull-down is not able to take into consideration. So for now, we think we have 100% foolproof way of detecting our RDF. Okay, so then the next question you want to ask really is, we are pulling out these different integrases and we're identifying the RDFs. How orthogonal are they? As you know, in synthetic biology, engineering biology, orthogonality, that is the ability of a system to be specific to its partner and not interfere with other systems, is a desired requirement. If you are using 4 or 5 different integrase RDF pairs, you don't really want them cross-reacting. And this is an initial AlphaFold multimer prediction of how orthogonal the different LSIs and the RDFs are. And of course, we need experiments to validate that. So that's our first challenge. How do you find RDF? And so we have been able to develop a system that allows us to successfully pull out RDF from a large number of candidate proteins, spending less money and spending less researchers' time in the lab. So that's a significant development. The initial draft of this manuscript is actually on bioarchive, and we hope we'll be able to get it through peer review in the near future. So our second challenge, which we try to address, is more of a basic science question. That's why are the RDFs so diverse in structure, yet they're able to interact with the integrase at the same interface? That is not the kind of thing you tend to see in biology. Structure and function tend to be conserved, or they co-evolve together. So how do you come about this system? So to be honest, people who have been working on this system in the past have always thought the serine integrase RDFs are monolighters. That is, they are proteins that have other primary functions, but somehow under stress or due to certain reasons, the phage have decided to hijack them, co-opt them, or repurpose them for a secondary function as RDF. But that is kind of hard to prove experimentally. So we thought we'd try and find out, if that is the case, what are the primary functions of these proteins. So what we did is that we took the first one, the RDF for phage BXB1, BXB1 integrase is well known, and used a protein structure comparison server, the DALI server. So essentially what you do is that you use AlphaFold2 to predict the structure of your protein and run it through DALI. And DALI will tell you if there is any known structure, any known protein that has a similar structure to what you're interested in. And so we did that, and the closest candidate is a 3'5' exonuclease that has some kind of structural similarity to a BXB1 RDF. So we thought, well, that's interesting, but it's even be more interesting if you can demonstrate exonuclease activity for our BXB1 RDF. So we designed some experiments, and long-suffering Alex, after several attempts, was able to demonstrate some activity that phage BXB1 RDF is actually an exonuclease in vitro. So that was interesting, that the protein truly is a monolighter. As an RDF, it has a primary function. The second one we tried is the RDF for phage TG1, which has a different structure. And again, DALI server tells us that it is very similar in structure to a different protein, a single-strand DNA binding protein. So again, we have to try and see if we can establish that. And what the binding assay here shows, that the RDF is actually able to successfully bind single-strand DNA, but has no affinity for double-strand DNA binding. So the kind of thing coming through from this is that the exonuclease and the single-strand binding protein are both phage replication proteins. So through a mechanism we don't understand yet, it appears that the phages are co-opting existing replication proteins and use them as RDF during the excision pathway. Now, you might think that's quite fundamental biology. What's that got to do with synthetic biology, engineering biology? Well, it shows that if you have an integrase and you're unable to find its RDF, there's a possibility we might be able to engineer the other proteins in the cell and turn it around and make it function as an RDF. And this is the model of what we think is happening. Because of time, I may not be able to go through the details of this, but we think if you imagine a phage ran into some difficulty and undergoes some recombination and lost its RDF gene as a recombinant phage, then that phage as a prophage is essentially stuck. It cannot catalyze an excision reaction. It can't go through the lytic process. The only way it's going to get out of that state is to look around other phage proteins and see if it can hijack or reconfigure an existing protein and turn it into an RDF. So it makes some changes, undergoes some mutation, and a new LSI-RDF will emerge. And we think that's the only way, really, that you can explain the diversity in the sequence and the structure of proteins that are used as RDF. Right, so the last point I'm going to talk about is the challenge of a full integrase RDF DNA complex, because as much as we would like to engineer these proteins to have a kind of bespoke sequence specificity, if we don't have a structure, if we don't know how it interacts with the DNA, we're never going to be able to do the kind of thing we are doing with CRISPR, where you can target CRISPR to any location using the guide RNA. So to be able to do that, we had to collaborate with our colleagues and friends in the US, Professor Phoebe Rice and her postdoc, Dr. Hyun Shin. And it is almost like the holy grail of serine integrase biology that people have been pursuing for the past 25 years. We actually have a full complex of the integrase, the RDF, and the DNA bound together before recombination and after recombination along the forward integration pathway. And we have the exact system in the excision pathway. So for the very first time, we have a complete picture of what happens during integration recombination and during excision recombination. Again, we put some of these data on bioarchives, and you may be able to review them yourself. Now, again, the question someone might ask is, of what significance is this fundamental concept to biotechnology or to bioengineering? Now, a key finding from this structure is that for the very first time, we can map how the different elements of the protein, the different DNA binding domain 1, binding domain 2, the linker region, the alpha region, what part of at B and what part of at P are they interacting with. Now, with that knowledge, we're in the prime position to begin to ask the question, if I have any specific DNA sequence in a genomic location, can I design or can I construct an integrase that will have the exact specificity to go into this location and mediate recombination reaction with minimal off-target activity? That's the aspiration anyway, and that's why we're so excited about this structure, that hopefully in the coming years we'll be able to combine the specificity known for CRISPR and the tidiness that integrases are known for to create the ultimate genome editor, where you can cut and rejoin DNA and do it specifically at the exact genomic location that you want to do it. So those are the three key challenges, really. And just to summarize, the 3 points I've kind of highlighted here is that, one, we've been using AlphaFold to help map the interface for integrase RDF interaction. We've verified that with mutations and biochemical characterization. We developed a virtual pull-down protocol that allows us to identify RDFs, thereby enhancing our toolkits for genome manipulation. We also learned that phages are likely repurposing existing proteins to use them as RDF. And finally, we're excited about the new cryo-EM structures that we believe will help us to model DNA sequence specificity. So, and just to acknowledge the different ones in Liverpool who have been supporting our project. Alex gave the first part of this talk, and Alex is here. And in Chicago, we have Professor Rice and her postdoc Hyun, who have been working hard to help us collaborate in addressing some of these questions. And finally, we want to thank the different funding bodies who have been supporting our work in recent years. And thank you for listening. I will be happy to take questions, either from myself or from Alex.
Okay, thank you. Thanks again, Femi and Alexandria. It's great to hear about the work you've been doing and the new unique features of the LSI recombination and the challenges you faced, and also how your transformative approach will help find RDFs and will help develop more flexible engineering and biotech toolkits. So sounds great, and hopefully look forward to some great work with industry ahead. So I think we've got about 3 minutes left. If there are any burning questions from everyone on the meeting today, please raise your hand or add things to the chat. Hi, Lucian. Hi.
Hi, thank you for it's really nice talk. It's not really my area, but it was very fascinating. Can I ask you a bit about the sizes of DNA, well, fragments that you can, I guess, interchange? So clearly they work with very large DNA pieces because they have to put in prophages. But like in your screening example, you showed there where you can swap between the two fluorescents. I mean, that fragment is clearly quite small. So that's also acceptable that you can have both the sites quite near each other.
Yes, thanks, Lou. So the at-base sites functionally is about 48 base pair actually, and the at-base sites a little bit shorter is about 40 base pair. So and it doesn't matter how big the payload is, you can have 25 kilobase. You just need the 40 to 50 base pair to be able to insert it into the genomic location you can use. And that's the advantage of the serine integrases. The tyrosine integrases tend to require bigger, about 115 base pair of recognition sequence, which of course puts some restriction on what you can do at a genomic location.
How close can the ATT site, the AT sites be to each other? Because I guess the actual protein complexes are quite big, right? So they can't really be literally, 10 bases apart, I guess.
Yeah, sure. So the challenge now is the problem of DNA topology. You've got to have, and so if it's intermolecular recombination, that doesn't matter. If the two DNA sites you're trying to recombine are on two different DNA molecules, it doesn't matter. But if it's within the genome, the minimum we've seen so far, we've tested so far, is about 100 base pair that we've looked at, but we haven't actually set out to ask the question, how close do the sites need to be? But I think we can get away with 100 base pair.
Thank you.
One final question from anyone? Okay. Well, we'll probably stop there then, and just to reiterate again our thanks to both Femi and Alexandria. We've had a great talk today. If you're on the call and you're interested in giving a talk at a future webinar, please do get in touch with the IBIC team. I'd also sort of like to remind you that we run these each month. In February, we'll be joined by two people from Imperigen to hear about how they're leveraging AI and robotics to transform enzyme development across industrial biotechnology, and to learn about their particular journey in building a successful tech bio company. I think the link to that webinar is in the chat. We also have several IBIC funding schemes open for those who are interested in getting their research from their labs out into the Northwest sector. And we've also got a conference coming up in June, so please look out for this on our mailing lists for registration opening in the next few weeks. And yeah, if you'd like to know more about IBIC and what we can offer, please do reach out. We've got a fantastic team here to support you, and a much wider team across the region who are all sort of working towards this. So please do get in touch. And I'd just like to thank everyone who's joined us today, and look forward to seeing you at a future one. Thank you very much. And thanks again, Femi and Alexandria.
Thank you. Good to see you, Rob.